Hi everyone, and thanks so much to Oxford Nanopore for the invitation to tell you about some of our work. So today we're going to talk a little bit about RNA isoforms and why they're really important, and then about how we and others have been developing single-cell methods to try and understand and study isoforms. And then I'll talk a bit about how, specifically in my lab, we've been trying to use this to look at brain development. So what are isoforms? So if we look at the figure and we imagine sort of that toy example of a gene that's got 5 exons, this is transcribed into a pre-mRNA, and then we have splicing take place. And so splicing removes intronic sequences and then sticks exons together. Now, where this gets sort of quite interesting is that one gene can produce many, both RNA and potentially protein products. And so a key process involved in this is called alternative splicing. And that's where the sequence that is exonic in any particular RNA can vary. And then what this means is that you can create alternative RNA products that can differ in their RNA function or regulation, and also create proteins that have different functions. And what this means is that both alternative splicing and the alternative isoforms that get made really have a key role in both many developmental and also disease processes. And so a key area of interest for our lab is trying to understand the molecular mechanisms that are involved in brain development. How does an organism go from an embryo or a zygote to actually form a structure or organ as complex as the brain? And this is really quite a central question in both neuroscience and in developmental biology. And trying to understand how brain development occurs is important in itself, but it's also really important for understanding what is sort of going wrong in both our neurodevelopmental and neuropsychiatric disorders. And so alternative splicing and alternative isoforms have a really key role in brain development. Now, it actually turns out that almost all human genes have alternative splicing and create multiple RNA products. And this is particularly the case in the brain, which has the most prominent use of alternative splicing of any human tissue. There's a whole heap of different processes that are driven or regulated by alternative splicing, and I've listed a couple here. This includes the differentiation of neurons from progenitor cells, includes neural migration, so that neurons can find where they need to go within the mature brain, and processes such as synapse formation. And so a question that we've had is, can we use single-cell RNA sequencing to profile isoforms in all the different brain cell types, and then find more isoforms to study that might be crucial to brain development? And so what is single-cell sequencing? I know many people will be familiar with it, but this is a pretty general audience, so we'll kind of just go through some of the basics. So when you do kind of more standard bulk RNA sequencing, you basically take your sample or your tissue or your group of cells or whatever it is, and you mash that up and extract the RNA out of that. And then when you measure the gene expression, what you're getting is an average of all of the cells that happen to be in your sample. So the key part about single-cell sequencing is we're not doing that averaging anymore. We are taking our cells, we're isolating them, we separate them from each other, and then we get a gene expression profile or an expression profile from each one of those cells. And this allows you to see a lot more information that was otherwise hidden when what you were doing was only taking an average. Now, a really key step technically when you do single-cell sequencing is your ability to know what cell each of your reads or sequencing reads came from. And this is done by barcoding, where each cell gets a unique barcode, so each RNA, and therefore read from that cell, has that barcode, so you can take your reads and associate them back with the original cell. And the reason why this is important, we'll talk about later. And so single-cell RNA sequencing has become super popular. It has a lot of different applications. You can use it to identify cell types and their expression profiles. So the image here is from a pretty early paper looking at retinal neurons. And the plot there, basically every single dot is a cell, and the different colors show you the different cell types. And so you can see, hey, look, there's lots of different cell types. You can also use it to look at developmental or disease trajectories. So another classic paper in the field from a few years ago, looking at zebrafish development. They took samples from a number of different stages on zebrafish development and were basically able to reconstruct the entire developmental trajectory. Now, the key limitation with their standard single-cell sequencing is you get gene expression information, but you get very little information about isoforms. And so a question we asked ourselves a few years ago is, how can we obtain this missing isoform information? And so us and others have, because obviously others have done this in the field as well, have then thought, okay, what we need to do is develop long-read single-cell methods so we can do isoform profiling, because then we can get away from just measuring, say, the 3' end of a gene and get full-length RNA transcripts. And so working with Matt Ritchie and collaborators in Melbourne, in 2021 we published FLITSeq and Flames. This was both a lab method and software to do long-read single-cell sequencing. At the time, it required match short-read sequencing in order to find those barcodes and work out which read came from which cell. Last year, we basically had an update to this with another piece of software called Blaze. And the key part about Blaze is now you can get your barcodes straight from the long reads. So you can do the entire single-cell, long-read single-cell process. There's no short reads required. You can do everything just with your long, in this case, nanopore reads. And so just quick information about Flames. It's a pretty software that you can use on quite a wide range of single-cell protocols. We usually use it for 10x, but it can be used on other protocols as well. You get your long reads. You can still do short reads if you want, but there isn't actually any need to. And then it will take in your reads. It will do what's called demultiplexing, where it works out which read comes from which cell. We then align it to the genome. We can get our gene counts. Then we also work out what are all the isoforms that are expressed in the sample. Then we quantify our isoforms, and now you've got both your gene quantification, your isoform quantification, and you can do all the fun downstream analysis that you want. Flames has been undergoing ongoing updates and development work. Just suffice to say that over the last couple of years, it's become faster, more accurate, and easier to use. It's available on Bioconductor, so if you want, download it, give it a whirl, and let us know any feedback. Okay, so back to the brain. So the particular part of the brain we've been looking at the development of is the cortex, which has the really amazing developmental process. And it's called an inside-out process, where the deeper layers of the cortex develop first, and then the upper layers progressively develop later. Basically, what happens is down at the very bottom, you have the ventricular zone and the subventricular zone. That's basically where all your stem cells are. And those stem cells, they divide. They create progenitor cells, such as radial glia, intermediate progenitor cells. And then the radial glia do something that's really cool. Basically, they reach their processes all the way up to what at the time is the top of the developing cortex. And then the developing neurons migrate up those processes, basically as a physical scaffold, to then populate each of the layers of the cortex as they develop. And so obviously, if you're studying humans, actual in vivo samples are difficult to come by, so a lot of people in the field have developed stem cell models of human brain development. These work really well because they recapitulate all the steps that happen in vivo development. So you start with stem cells, you then develop neural progenitors, these divide, and then you get the deep layer neurons forming first, and then the upper layer neurons forming last. And so to show you some pictures of what it looks like, this is some day 25 where you have mostly neural progenitors. If you can see in the red, you get these little kind of circular clusters. These are called neural rosettes, and these are the developing progenitors and really early neurons. You get to about day 55, and what actually happens is the neurons quite like to hang out with their friends. So if you see in blue, you get these clusters of neurons, and then in green, we've marked all their axons and processes, and you can see that they just extend all these processes out, and then they start forming synapses and communicating with each other. You can also see this with light microscopy, where you can see all of the axonic tracks that the neurons are forming. And so what we did is we utilized this brain developmental model process, and we did single-cell sequencing from 4 different time points. We've got our stem cells, we've got our neural progenitors, we've got day 55 where you start getting more of the deep layer neurons, and then a later time point where you got more mature neurons are forming. We did Promethean sequencing with at least one Promethean flow cell per sample, and then we did the Blaze Flames procedure. So what did we do? So for our 8 samples, we have over 600 million reads, and we've got an average depth of about 30,000 reads per cell. The reason for this is that we want to have the ability to actually be quite sensitive in detecting and quantifying isoforms. And what we're able to do is get, compared to sort of some other methods, quite a large number of isoforms and genes per cell. Overall, we identified about 130,000 isoforms. Most of these were known, and around 6% were novel. Now, this is a bit of a lower percentage of novel isoforms than you see in some of the other results out there. We believe and we know this is quite conservative, but what we're really keen to do is try and avoid getting false positive novel isoforms, because studying those doesn't get you anywhere. So we're really trying to make sure we're emphasizing signal. You can see this in another way using the squanti categories, where about 90% of our isoforms are what's called a full splice match or a complete match to a known isoform. We have almost no, basically known isoforms that are incomplete. That's that ISM category. And then we have about 10% that are in the various novel categories. So you've got novel splice isoforms, novel intergenic RNAs, etc. And you can obviously do some interesting things like ask, how many isoforms actually, even just in the process of neural development, does a gene express? And what you can see is the vast majority of genes are expressing multiple isoforms, even within this just particular system, and quite a lot of them are expressing, a very large number of isoforms. We then wanted to have a look at what cell types are present. I'll just emphasize we're only using long reads here. There's no short reads to identify cell types. And so this is a UMAP plot, similar to the one I mentioned earlier. Each little dot represents a cell. Each color represents a different cell type. And what we can see is we've got our stem cell cluster. We've got our progenitors cluster, so we can see those radial glia and intermediate progenitor cells I mentioned earlier. And then we've got about 5 different types of neurons that we can see. We then wanted to see if we could find a developmental trajectory within the starter, and so we've done this in a couple of different ways, but suffice to say that it lines up really well with what we'd hope and expect, in that the earliest samples are the stem cells, and then you go through your neural progenitors and into your various types of neurons. So that works really nicely. We've then looked at differentially expressed both genes and isoforms between our different cell types, and we can see obviously many hundreds of these. Again, this is a fairly conservative measure. But when we look at what some of the genes which are differentially expressed are, it looks really good. For example, we see OCT4 as one of the differentially expressed genes in stem cells. This is a classic stem cell marker of pluripotency. Our radial glia express VIM, which again, classic marker of radial glia, along with a bunch of histone proteins, which tell us these are a rapidly dividing kind of cell. And then when we look in our neurons, we see things like calcium channels, we see the glutamate receptor, and we see other classic neuron genes. So we know that these genes are doing neural development, they're starting to form synapses, etc. When we do a gene ontology analysis to have a look for what sort of processes are enriched in some of our cell types, so this is just taking the intermediate progenitor cells, I think if you look at that, you can very clearly see that we're getting a real strong signal of proliferation. Whereas if we take our neurons, we see a strong signal of neuronal development, and we see a lot around about, say, like the receptors and the ion channels, and when you've got developing synapses. Okay, so I'm just going to finish now talking about a few examples of some of the interesting and some novel isoforms that we're seeing. So I'm going to talk a little bit about a gene called PKM. So it's a pyruvate kinase, and if you're sort of someone like me and you get more excited about seeing a splicing factor or a transcription factor, a metabolic enzyme might seem not quite so cool, so apologies to all the metabolism people out there. But what's kind of interesting about PKM is it's known to have multiple isoforms. And some of these isoforms don't just have metabolic functions, they actually are reported to be able to go into the nucleus and act as transcriptional regulators. So it actually seems to be both a metabolic enzyme and a transcription factor, depending on the isoforms that are being made. Now, when you do gene level counts, which we obviously have in our data, it basically looks the same across all the cells in our data set. However, when you go and do isoforms, you see something that looks a little bit different. Now, I'm just going to take a second to explain this image here. This is from a tool called Isoviz, and it allows you to visualize both your isoforms and their expression. And so basically just similar to kind of like a UCSC genome browser, all of the black boxes are exons, all of the lines joining them are introns, and then up the top in the purple are the protein domains which are mapped to each exon. So you can sort of understand how isoforms differ both in their exonic content and in their protein domains. Now, we see a novel isoform of this PKM. You can see that it's got a novel exon, which will look to us to probably either add to or disrupt one of the pyruvate kinase domains. When we look at which cells express which isoform, we see something quite interesting. I'll just note that in this UMAP, because of the way UMAP tends to sometimes behave, it's flipped things around. So the stem cells are now on the right, the progenitors are on the bottom, and then on the top is the more mature neurons. What we see is that despite the fact that the gene is basically uniformly expressed across all the cells, this novel isoform is basically only expressed in the neurons, whereas, and you can also see that here, whereas the known isoforms are the known isoforms are basically almost entirely expressed in the progenitors. So we've got something quite cool here. We've got a novel isoform switch, and so it's going to be really interesting to sort of follow this up and try and understand what the different functions of these isoforms are and why some of them are only in progenitors versus another one is in the mature neurons. Okay, here's a second example. So this is another gene called GPM6B. So it's a membrane glycoprotein. It is putatively involved in brain development, but it's not really functionally characterized in brain development at all. So again, we're able to see a novel isoform of this gene, as well as some known isoforms. So there's quite a lot of transcriptional complexity for this gene during the brain development. When we look at the known isoforms, we see that these are preferentially expressed in the progenitor cells. In contrast to what we saw with the PKM gene, in this case, the novel isoform seems to be expressed in all of the cells of both progenitors and the developing and more mature neurons. So we've got kind of the opposite here, where actually it looks like the novel isoform is probably the dominant isoform of this gene and seems to be fairly constitutively expressed, whereas the known ones are perhaps often more lowly expressed and actually more cell type specific. So again, it'll be really interesting to now follow this up and understand what the different functions are and why they're doing what they're doing. Okay, so just to summarize, we've put in quite a lot of work to help create software tools to analyze long-read single-cell data, and all of the work I've shown you and all of the data is completely without any short reads at all. This is all based just off your long reads. We've used this to try and better understand in an ongoing project the role of isoforms in human brain development. And obviously with this we can do things like we can see known and novel isoforms, we can detect cell types and their trajectories, we can quantify isoform expression, and we can see switches between cell types and overdevelopment. So I hope this also convinces you that this sort of technique with the long-read single cell, however you're doing it, really has a lot of promise if you're interested in isoform dynamics, not just in this system, but in almost any system that you're interested in, because you can get some really detailed understanding of what isoforms are being expressed in each cell type. And so I'd just like to finish by acknowledging the people who've actually done the work. So within my lab, Sefi, Upay, and Manvier are our long-read single-cell team. Also want to acknowledge Matt Ritchie and his team at WeHi, who have been sort of the main drivers behind the initial version of Flames. Also collaborators at University of Melbourne, the Shim Lab, who we developed Blaze with, and Claire Parrish and her lab at the Florey, who we worked with to do all the neuronal differentiations. And just thanks everyone for listening.